Good morning, children. How are you, children? I think you all are fine. Yet, class students, we successfully completed our first chapter, What is Science? Now, you have idea about science. What is science? Yes or not? Yes. Today, we are discussing our second chapter, the cell, the basic unit of life. Today we are discussing about our next chapter, the cell, the basic unit of life. Our earth is a wonderful place where so many beautiful creatures lives very happily. Yes or no? Yes, our earth is very beautiful. We observe beautiful green plants and brighten flowers, huge animals like whales and smaller creatures such as insects, ants. Yes or no? Yes. We can't imagine our earth without life. All these living creatures makes the earth very joyful. You know, children, some of the creatures also present on the earth, but we can't observe them with our unaided eye. We can see the creatures that are visible to our eyes. But some of the creatures also exist in the earth that cannot be seen through our unaided eye. మనకు కంటి కనిపించని కొన్ని జీవులు కూడా ఉన్నాయి. Everyone think like this only. When you go back 350 years ago, because the effort of three major scientists, we came to know about the understanding of these unknown creatures. Okay. The organisms that can be seen through only the microscope are called as microorganisms. Nowadays, we know lot of information about the microorganisms. Yes, correct or not? Yes, we know lot of information about this microorganisms. But whose effort is there? Because of the three major scientists, who is nothing but Athanasius Kircher. The first credit goes to that one, Athanasius Kircher. Next, Jan Swammerdam. Next, the third very important person, is Anton von Leeuwenhoek. Who? The first person is Athanasius Kircher. The second person is Jan Swammerdam. The third famous scientist is Anton von Leeuwenhoek. Among them, the effort of Anton von Leeuwenhoek are appreciable. Why? Because nothing but he first time observed the microorganisms by using the magnifying glasses. In our general language, bhutaddam. Your children feel very happy while you are playing with that magnifying glasses. Yes, na children? Yes. Same, he also first time observed the microorganisms by using this magnifying glasses. He first time put the waste material of teeth, means in our language palla pachi, under the magnifying glass, and he observed that some of the creatures are actively moving, means nothing but while he observing the waste material of teeth, he observed that some of the creatures are moving very actively. He named them as animalcules. Means nothing but animalcule means nothing but smaller animals only, but he observed them in a very smaller size that cannot be observed through our normal eye vision. So that's why he gave the name as animalcules. So it leads to observe the microorganisms. Means his experiments encourage to the other scientists to perform their experiments continuously to know further information about the microorganisms. Then later, in the year 1665, the scientist named as Robert Hooke observed the cell in the cork of oak tree. Cork means nothing but beradu. Okay. At the time, he observed honeycomb-like structure. Means hexagonal shape like structures. And he gave the name to that structure as cell. So, in Latin language, the meaning of cell is little room. What? Little room. In Latin language, the meaning of cell is a little room. So, the oak trees are not grown in our surroundings. మన ప్రాంతంలో ఓక్ చెట్లు పెరుగుతాయా? లేదు. So, to observe similar structure that can be observed by the Robert Hooke, we should choose the matchsticks. means aggi pullalu. Why? Because nothing but that are also made up of with strong wood. అంటే గట్టి కట్టెతోనే చేయబడతాయి. Which one? Mastics. So first of all we know about the activity. What is the aim of that activity? Is nothing but to observe the cells of the mastic under the microscope. Listen carefully, children. What is the aim of that activity? To observe the cells of the mastic under the microscope. Okay, what is the aim of that activity? To observe the cells of the mastic under the microscope. Material required. What material should be required to perform this activity? So mastics. What? Mastics. Watch glass. Watch glass means it is similar as saucer. You know, cup and saucer. Same, but it is made up of glass. So, watch, that is called as watch glass. Next, glass slide. You know, mirror strip. That glass slide also same as mirror strip, but mirror strip one side is coated with the one of the material, but glass slide is completely transparent. So the materials are mastics, watch glass, glass slide, water, and blade. What? Blade, glycerine, cover slip, compound microscope. What is the material required? Mastics, watch glass, glass slide, water, glycerine, cover slip, compound microscope. Procedure. Normally the mastics are very hard material. అంటే చాలా గట్టిగా ఉంటాయి. Normally the mastic are very hard material. So we should soak the mastics in the watch glass that contains water for about half an hour. Generally the mastics are very hard material, so we should soak the mastics in a watch glass that contains water for half an hour. After half an hour, remove the mastics from the watch glass and make thin slices of mastic. అంటే చిన్న చిన్న ముక్కలుగా చేయాలి అగ్గి పుల్లని, and make very thin slices of mastic. Now choose a thin section of mastic and place it upon the glass slide by adding a drop of water. Choose a thin section of mastic and place it upon the glass slide by adding a drop of water. Now add 1 drop of glycerine to the slide. Cover the slide with cover slip. Cover the slide with cover slip. Place the slide under the microscope and observe the slide under the microscope. Okay, once again procedure. Generally the mastic is made up of hard wood, so we should place the mastic in a watch glass that contains water for about half an hour. Within a half an hour time, the mastic is completely soaked in the water. Now carefully remove the mastic from the watch glass and make thin slices of mastic. Place one of the thin slice of mastic on the glass slide by adding a drop of water. Now add one drop of glycerin. Cover the slide with the cover slip. Place the slide under the microscope and observe the cells of the mastic under the microscope. Result, we observe hexagonal shape structures under the microscope. These are the cells of the mastic. Okay, children, with this we completed this activity. Okay, how do you observe the cells of the mastic under the microscope? Okay, you should properly write aim, material required, procedure and result. Thank you, children.